So please accept if you wish to continue. Please share my screen. So hello all again. Thank you for joining us today again. Pleasure to see you all. Once again, my name is Krishna. I work at the Gladstone Institute's Bioinformatics core. I hope you all were there yesterday. In any case, I will start with quickly reviewing what we discussed yesterday, and then we will pick up from our discussion of normalization, etc. And also we will be looking at the script just as we did yesterday. Once again, our goal in this workshop is to broadly discuss the methods for analysis and the various algorithms, and also get some hands-on experience with running one specific software package, for analysis of the single-cell RNA-seq data, such as SURAT. So that's what we are going to be that's what we'll continue to do today. And as we discussed yesterday, I think you all perhaps know bulk RNA-seq assay well, and we discussed it yesterday, so I'll just skip it. But if you have any question, I will scroll through the slides, moderate pace. If you have any questions regarding anything that you see on the slides that you did not get a chance to ask yesterday, or you want to revisit today again, please feel free to unmute yourself and ask at any point in time. So once again, the way single cell technology works, we are again going to single cell technologies again rely on DNA sequencing, high throughput sequencing instruments such as from Illumina to capture the sequence of DNA fragments. But the way DNA fragments are generated, there is a procedure by which fragments can be tagged with a barcode that will help us assign groups of fragments to a particular cell. So that's what is being enabled by single cell technology. And the way it happens is there is a instrument which utilizes, my understanding is it utilizes a microfluidic device where one loads cells along with some other enzymes and other mix of nucleotides, etc., at specified levels of dilution. And also there are some beads which have DNA oligonucleotides sticking out from them, and these beads have barcodes and unique molecule identifiers, along with priming sequences and adapters, which will be added onto the mRNA sequence that will be captured in the cDNA form upon reverse transcription. So these cells, they are encapsulated in drops, nanoliter sized drops with beads, which have these oligos, and then the reactions of reverse transcription happen within the drops, so that the mRNAs within a cell, they get tagged with the barcode, which will be a unique barcode for each cell. And then each molecule gets a unique molecule identifier so that we can deduplicate PCR duplicates when we do after amplification. And again, we discussed that droplet-based technologies are dominant in the field because of their high throughput. And we discussed some of these things also regarding the limitations and applications of single-cell RNA-seq. So I'll skip through that unless you have some questions. Just to also remind you how the data is structured, we have the output from pre-processing of the FASTQ files is a counts matrix for each sample, counts matrix for our data. The counts matrix has rows representing the genes, and if there are, let's say, 20,000 genes which you have observed in one or the other cell, then there will be 20,000 rows. And if you have 6,000 cells, or if you have 6,000, then there will be one column per cell. And if there are 6,000 cells per sample, then you will have multiple samples. And let's say you have 2 samples, so there will be 12,000 columns. So that is the kind of data we have. It is, and also we noted yesterday that one characteristic of the data, in contrast to bulk RNA-seq, here we have a lot of zeros. And that is because our sequencing is distributed over a lot of cells. And if we want, let's say, 1,000,000 reads per cell, then if we have 6,000 cells, then we will be looking at lots of reads, and that is expensive. So typically, the typical sequencing depth that people go for, it leads to lots of zeros in the counts matrix. We discussed the kind of a standard workflow for analysis of single cell RNA-seq data. After one has done their experiment design, library preparation, sequencing, the data comes in, and then you have to demultiplex, align the reads to a reference sequence, and also do counting of where the reads are mapping to. So which genes are getting how many unique molecules associated with them. So you get that in a counts matrix, and then you start with analysis in, let's say, R or Python or Galaxy using scRNA, or what we are doing today, using scRNA for the analysis in R. But you can also opt for, let's say, ScanPy in Python or Galaxy or other things. And some of the steps which are common to all of these different tools, including bioconductor, suite in R. There are lots of packages in bioconductor also, using which you can implement these approaches or these steps of analysis. So to start with, you will have to do quality control of the data to check if there are any cells which are perhaps more, if there are any barcodes which are perhaps more like, associated with empty drops, or let's say doublets of cells, then we will filter them. Then by the raw counts are not normalized, in the sense that we cannot compare the raw counts in one cell for a gene, the raw counts for a gene in a cell with the raw counts in another cell directly without normalization, because things, the raw counts depend on the sequencing depth for each cell, or the total number of molecules observed in a cell, which can vary from cell to cell. So there is a need for normalization, and then we will pick up with our discussion on feature selection and dimensionality reduction and the other things. So I will scroll through these slides. If you have any question, please feel free to stop me. We discussed this barcode rank plot, and I showed you how things can look if the data is poor quality, or if lots of our cells, for example, we saw that the barcode rank plots can be more rounded if the sample was compromised in some way, where we cannot identify which are empty drops versus cell-associated barcodes, or there can be multiple plateaus kind of, or levels of UMI counts in the cells if the sample is very heterogeneous, or if there are two kinds of cells. So those kinds of things can happen, and we'll discuss these things briefly. Again, if you have any questions, please feel free to ask. Also pointed out some of the considerations when you are doing pre-processing, such as filtering or not filtering of count matrices. Cell Ranger, if you are running Cell Ranger for pre-processing, it will output both. And well, depending on what tools you will choose in later steps of analysis, you may want to work with filtered versus unfiltered matrix. And also regarding sampling, we discussed that you have the option to set it to either downsample the reads to make the total number of reads per sample equal, or not downsample, so you can do that. And we also discussed that the latest development from the developers of scRNA, which is the SC transform method for normalization, it recommends not downsampling. So, but that is not true for all the methods out there. So you may still need to downsample, but for some of the analysis steps, you may do without downsampling. And we also looked at the data, how it is organized in 3 files. The data is generally stored in a matrix market exchange format for sparse matrices. And We looked at that yesterday. So if you have any question again, please feel free to ask. Otherwise, I will skip through this. And we looked at various considerations for quality control and filtering of cells based on quality metrics. So hopefully that all made sense yesterday. And this is what we discussed last. We discussed that the raw data by itself is not normalized. And the first thing that comes to mind is dividing by the total UMI counts in a cell. So for example, if you want to normalize the raw counts, or compare the counts for one gene with another gene, then one approach that comes to mind is you divide by the total UMI counts in a cell. Then maybe one can assume that or one can expect that the or one would expect that the effect of total sequencing depth variation from cell to cell will be accounted for, but that is not the case, as we looked at with this figure, because one would expect that after normalization, the expression level of genes, if they are appropriately captured by the relative counts, then they won't vary with the total cell UMI count. But they do vary for some of the categories of genes, at least for this particular data set that the authors examined. But in general, they could vary for all of the categories. So raw dividing by total sequencing depth is not a good practice. And we also discussed log normalization, which is the default practice in the field right now, including in the scRNA pipeline. The authors of this is from the same lab that is behind the scRNA package. And what they found was that log normalization is not also leading to that desired goal of independence of normalized expression values versus the total cell UMI count. We still see trends which are not ideal, and they can lead to false discoveries of or false positives in our steps of differential analysis. For example, these authors compared a data set with its own downsampled version, and they found that they were still greater than 2000 DE genes after log normalization. So that means that log normalization is not it cannot account for the variation in total number of UMIs per cell, or it does not account for it. And we discussed this approach of assay transform briefly, but what we get, I will not review the whole thing unless you have some specific questions regarding what we discussed yesterday, in which case please feel free to unmute yourself and ask. I will point out a couple of things. One is that after normalization, what you get here from assay transform is values that range from negative values to positive values, and the mean value for each gene, I believe it should be 0. Yeah, I think it is definitely 0 for each gene. And the interpretation is after normalization, what you get the values, if they are positive, it means that the observed level of expression of a gene in a certain cell is above what would be expected given its mean expression in the population that you are looking at, and given the sequencing depth for that particular cell. So that is the interpretation of positive normalized values, and the negative values would mean the opposite, that the observed expression is lower than what would be expected given its mean expression in the population and the total number of UMI counts in the cell. I hope that makes sense. And also, one can look at the variance of the normalized values, and the theoretical expectation for the variance of genes which are not differentially expressed across the population is that they will have a variance of 1 or distributed around 1. but genes which some genes may have very high variance, which will be indicative of something, some biological phenomena affecting their, some factors affecting their expression levels in certain cells, which could be disease, in certain cell that may be impacting certain cell types and the expression of certain genes in those cells. So we also ran, I hope you all had a chance to look at the script in some more detail yesterday. We ran the step of normalization yesterday, and I will just point out that if you look at the documentation for normalized data, the function which is doing the normalization, it provides three approaches for normalization. One is log normalize, which we discussed, and that is the default practice in this function, log normalize. But there are two other approaches implemented. Relative counts is just dividing by the total UMI counts and multiplying by 1,000,000 to convert it into counts per million. And or perhaps it does not do that. It is multiplied by 10 to the power 5, but if you multiply by 10 to the power 6, then you will get counts per million after dividing by the total UMI counts. But that is not the recommended practice. It is still an available option. And the third approach, the CLR approach, is what we want to, let's start picking up from there, our discussion today. So besides, scRNA is a package which allows you to analyze lots of, well, they are, building it to be general purpose, as I understand, for single cell genomics. And within the umbrella of single cell genomics, there are, single cell RNA-seq is one kind of thing people are looking at, but then there are also proteins which you can pursue in your studies. And then there are other things also. So for, if you are working with, there is at least one assay which is called a site-seq. So site-seq allows you to capture also the abundance of surface proteins in cells. And the way it is done is using some antibody conjugates, complexes of antibodies with oligonucleotides. So they have engineered these site-seq folks. They have engineered these antibodies, oligonucleotide conjugates, and there are a bunch of antibodies which are available from their, I don't know who is supplying them, but I guess the lab or the vendor or someone. But these antibodies, they can target some of the surface proteins in certain kinds of cells, where they are expressed. And so if you incubate your cells with these antibodies, then the antibodies will latch on to the surface proteins, and with them will come an oligonucleotide, which is specific to each antibody. So, and with these antibodies, you can tag your surface protein with oligonucleotide, which can be sequenced in as just like any mRNA or anything else, inside the cells. So that's how it works. But there are typically, if you are doing this assay, where you will get mRNAs, but also for the surface proteins, you will have tens of surface proteins for which you will have information at the single cell level. But the challenges of normalization in this case are a little bit of a different nature, I guess. So one challenge that comes up is with due to the problem of what is called as compositional data, which we can understand hopefully with these three tables that I have in illustration at the bottom. So let's say there are three proteins which you are looking at using the site-seq assay. So, well, the three proteins, maybe their expression levels or their abundance on the surface may be varying independently. But if you just divide by total counts for all the proteins, then you may see some spurious correlations which are not actually there. So for example, here, the gene or the protein in row 1, its expression level is varying hugely in the three samples that I have illustrated here, while the other two not varying so much, and they are, wherever they are varying independently. For example, from sample 1 to sample 2, the row 2 goes down, while row 3 goes up, and something like that also in sample 3. However, when you look at the relative counts, it looks like the row 2 and row 3, they are simultaneously going down or simultaneously going up because of the way relative count works. Everything has to sum up to 1. So if one number is dominating, or something cannot increase without decreasing another value in the table after, if everything has to sum to 1. And this constraint of all the numbers summing up to 1 for the total expression of proteins is not something that is generally accounted for in some of the downstream steps for analysis. So to account for that, to make sure that there is no problem downstream, your normalization should not be using relative counts. That is not appropriate for this kind of data. And for that, there is this centered log ratio approach, which you can read about in your own time. I just wanted to mention, if you are trying to choose the normalization approach, then this CLR is not for single cell RNA-seq data. It is there for proteomic or protein information, abundance information, which people are also looking at along with single cell RNA-seq in some of the studies. Any question regarding that? I hope I was able to make myself clear or convey the key point here. There are also other methods available for normalization, such as bioconductor package. I believe this is a bioconductor package, scRNA, which allows for estimation of size factors in the sense that people do in bulk RNA-seq. Some folks also have been working on normalization approaches using spike-ins. So there are different approaches, but in SURAT, the default is log normalization, and recommendation is assay transform. So you can look at SURAT vignettes, and you will find how to run assay transform, which is basically, instead of running normalized data, you will run a function from assay transform, which is also called as assay transform. Any questions regarding normalization? We kind of went a little bit fast on that topic yesterday. So I just want to give you all an opportunity if you have any question on that. There will be more discussion on this on Wednesday, including with differential analysis and batch correction. So you can also ask your questions then, which is tomorrow. Okay, so well, thus far what we have done is we have done some quality control of the data, and we have hopefully factored out or accounted for, or converted our raw counts to something, to values which are more representative of the expression level of genes in the cells. And now the next goal is, or the next step is to do feature selection. So first of all, why work with a select set of features? There are a few reasons for that. One is that our expectation is that, if you have properly designed your experiment and or studying something, with a proper experiment design, then all the genes will not be varying simultaneously in your population. Maybe a subset of genes out of 20,000 plus genes will be varying or will be of interest to your study. So we want to narrow down our focus. If we work with all the genes that are present in our population, our data, then, well, there may be some set of genes which are varying in a noisy way. They are varying just because of intrinsic biological noise, but not something related to the phenomena that you may be studying. So we can perhaps get rid of those subsets of genes, so to say. So, and by doing that, we can improve the signal level in our remaining data. So that's what we want to do: enrich for signal content, or the desired signal content in our data. So that is the goal of feature selection. And it may also improve computational efficiency, because we will be reducing the size of our data for downstream steps. so it can improve computational efficiency. So those are the reasons to do feature selection. And now then, it raises the question of how do you select those features? And again, for that, there are various practices, but the most common, at least in my perception or experience, is that people are doing selection for highly variable genes, which means that you look at the variance of genes, the normalized expression values, and you select the genes which show the highest levels of you select the top 2000 or 3000 or 500, a certain number of genes which you specify. You select that number of genes which show the highest magnitudes of standardized variance, um, not the variance of the normalized values themselves. And the reason to do that is that different genes, like if some genes are very highly expressed and they have 10% relative, let's say, error or noise in their expression levels across the population, and some gene is very poorly expressed, then in absolute terms, if the 10% relative noise in the expression level of very high expressed gene will be, in absolute terms, will have a much higher value than that of the poorly expressed gene. So you have to standardize the variance so that you can select them all on the same scale. The variances are kind of on the same scale and kind of account for the difference in the expression levels of different genes also. So that is what the standardization is doing. ST transform, if you are running ST transform, then the normalized Pearson, the normalized expression level from ST transform, you can take the variance of that, and that is already the standardized variance. That is my understanding. But if you are working with log normalization, then there is a similar in spirit approach for to assess standardized variance. Let's just look at this equation here. So once again, if you are working, if xij's were instead of them being like if instead of calculating the normalized expression from SC transform, let's say we are using log normalization, then once again we will use the mean of the log of the mean UMI counts for the gene and divide it with the predicted variance or predicted standard deviation for the gene given its mean log normalized UMI counts. And that prediction is based on, again, a curve fitting kind of approach where you have, for each gene, you have its mean value, the mean log normalized UMI counts, and also its variance. And you look at that for each of the genes in the population, and then you fit a trend line. And then the trend line is the prediction for each gene's variance. So the standardized variance also for log normalization works in a similar fashion, but again, there are differences because of the way data is normalized. So this figure on the right is illustrating the selection of the genes. And if you have any question, please ask now. I hope I'm making sense. I won't know unless someone lets me know that I'm not making sense. So I'm happy to repeat whatever I've said. But someone has to say that I did not make sense. So, okay. Well, I hope you all have in your session steps until normalized data and the data object available. If not, then you can run it until this chunk. I think if you hit this button, I think it will run all chunks above and until this. So you can do that. I will run this chunk for me. What we are doing here is, first of all, we are using the function find variable features. We are passing it the surat object that we created yesterday with the normalized also counts in there. And then we are asking it to perform the standardization of the variance. using the variance stabilizing transform, which I just described. So here we use log normalization. So we have to perform this standardization, and then we are asking it to select 2000 top highly variable genes for our next steps. So that's what it is doing. And we get these plots in the next steps. So let me go to the markdown output. So we here, I have printed some of those top genes. Again, I'm not really into these genes, so I don't know what they are about. And we can look at these plots if we want to see how much those standardized variances differ from the bulk of the distribution, things like that. We can compare that. Any questions about the code itself, or I think the code is self-explanatory if you understand how functions work? Or if about the feature selection? Well, once again, all questions are welcome. There are no stupid questions, only stupid answers. So I'm happy to answer any questions that you may have. I will also go ahead and run this next chunk of code, which is about dimensionality reduction. It takes a bit of time. So if you have, if you don't mind, you can just run these next chunks. Of course, I have the markdown, the HTML file also with all the plots there, so we can discuss them. But while we look at the slides, I will just run them here. So the next discussion, before we actually move to the next discussion, I also perhaps just want to point out that there are other selections of other ways to select other ways to select features, which is again referring to genes in this case. So besides looking at the highly variable genes, some authors have recommended selecting genes based on domain knowledge. I have not seen that selection very commonly, and one of the reasons perhaps is that if you write something where you have say that I have selected my own set of features based on my own domain knowledge, people may ask questions about the subjectivity of the choice, etc. So that's there. And another thing is another way to select features is based on genes, based on the dropout rate of a gene. So we discussed that each gene will have zeros in some zero observed value in some cells. So some authors have also discussed selecting genes based on what fraction of cells have no counts associated with them, and if that fraction is higher is very high compared to what is expected given the expression level of that gene, then they rank the genes by this how much they deviate from the expected, how much their dropout rate deviates from the expected value, and select based on that. And then there are other approaches such as other kinds of models to select highly deviant genes and etc. We will perhaps discuss this in the next section about highly deviant genes. So there is a question from Paul Su. Thank you for the question again. So when you their question is, so when you run SC transform, you do not need to do variable feature selection. Yes, that is correct. So when you are running SC transform, let me see. Then the SC transform step of running SC transform, it itself selects the features of interest. And after that, you can straight jump on to dimensionality reduction. So feature selection is happening. If you are running SC transform, it is happening within this function itself. There is one function which is doing both. Let's see if this is not finished. We can look at that. That's a good question. That's what I was also going for. So it is selecting 3000 features by default, which you can change variable features dot n, I believe that is what it stands for. So yeah, it is selecting 3000 features based on the variance of the residuals as I described yesterday. I hope that answers your question. Any other questions? Okay, well, so now if you don't have any question, we can get started with, I guess, our next section of dimensionality reduction. So first of all, what are dimensions? So let's say we perform an experiment where we are dealing with only 2 genes at a time. Then we can plot ourselves, or we can visualize ourselves in 2D, where each of the axes will represent one of the genes, and our points here will be one cell, and we can visualize our data in 2 dimensions. However, we have done our experiment in a single cell RNA-seq experiment. We capture tens of thousands of genes, or 20,000 or 30,000, depending on how large our genome is and how many genes there are. So we have thousands of genes, and well, now we have a very human limitation. We cannot visualize more than 2 or 3. We cannot visualize more than 3D at a time. So now, if we are working on visualizing, if we need to visualize data, which we do need to, to make sense of it, then we can't really visualize all of the dimensions simultaneously. So we have to reduce them maybe. And there may be other goals also. For example, reducing dimensions may denoise part of the data, may lead to denoising of the data. It may serve that goal, but also increase the perhaps computational efficiency for some of our steps. So there are various goals, including, as I mentioned, denoising, computational efficiency, data visualization. These are some of the goals for reducing the dimensions from 20,000 to fewer. So there are at least 2 kinds of dimensionality reduction algorithms which are applied for single cell RNA-seq data. One is called as linear approaches or linear reduction, and the another is or linear projection, and another is nonlinear projection or nonlinear dimensionality reduction. So we will discuss that briefly in this section. And first of all, let's discuss the linear projection method. So if we have, let's say, if we are in our sample, we have 2 kinds of cells, disease cells and healthy cells, and that is what we have experimented with. So let's say, these samples or these cells are distinguished by the expression of one gene, like let's say gene 1, it is, let's say, the gene associated with the disease. So, and let's say the diseased cells have a lot more expression of this particular gene than the healthy cells. While the gene 2, it really is not associated with the gene. We did our experiment with this gene, but it's not associated with the disease. And whatever variance or variability in the expression level of this particular gene we see in our data, that is maybe, let's say, driven by intrinsic biological noise and not the factor of disease. So, well, in that case, we may find that our data is distributed something like this, where the healthy cells have very low value for gene 1 and high value for gene 2, while very low value for the healthy cells, gene 1, and very high values for the disease cells for the gene 1, while along the axis of the gene 2, we don't see any relevant or useful variance or spread. This is just noise in the direction of gene 2. While if gene 2 were associated with the disease, then we may find that our data is distributed something like this, where there is, let's say again, the healthy cell has low expression of gene 2, the disease cell has high expression of gene 2, while in the direction of gene 1, there is noisy spread, nothing associated with the factor of disease. Both the two clusters are actually spread along in this direction. So that's what I mean by noisy spread. In reality, though, it will never be, perhaps, or it may be, but it will seldom be that only one gene is associated with the whatever factors or biological phenomena we are studying. It is generally a set of genes, maybe, which are together participating in some pathway, or maybe they are associated by some go term or something. There is some biological phenomena which associates a bunch of genes together, and that biological subsystem of our overall cells are perturbed by in the presence of disease or whatever else we may be studying. So in that case, what we will see is that our cells are actually spread or stretched out, in a composite direction of multiple genes, not just any one gene, but many genes. And you can also, extend this, if you can, in your imagination, to very high dimensions, thousands of dimensions. And then in very high dimensions, well, we cannot, really see it, but you can see that the data will be stretched in certain directions much more than in other directions. And those directions in which it is spread much more, they will be in the direction of some of the genes, or they will be composite directions made up of contributions from the various genes contributing to, let's say, the pathways associated with the disease. So when we are doing the linear projection, what we want to do is reorient our data such that we are looking at the data from the perspective of these hidden factors, which are associated with, again, let's say, the pathways that are altered or perturbed in the presence of the disease. Does that make sense? A few affirmations or confirmations will help, but it's all cool if you all just want to listen. That's all good. So in any case, what we want to do is ascertain if there are these directions or these hidden factors present in our data, because they may also serve as a quality control. Maybe like their presence indicates that there is something to worth proceeding for. If they are not there, well, then maybe you need to redo the experiment instead of doing any analysis further. So that's what we want to do, and I will explain it in more slides. But how do we identify those directions? So there are linear algebra techniques, which we will not get into, but the method is called as a principal component analysis, or PCA in short, and you will find it being applied in a variety of analysis in genomics and bioinformatics. It's very common, and you may have already seen it in plots in some of your papers, some of the papers that you may have been reading, or applied it in bulk RNA-seq analysis. So in bulk RNA-seq, for example, we typically see that there are one dimension. If the experiment has worked well, then we will see that one of the dimensions is associated with the disease associated pathway or something, and the second dimension associated with hidden factors such as batch effect. Another hidden factor, example, batch effect, but it could be something else, like variation of age among the samples that you have studied. Here we have single cells, and the single cells may also be spread in those in the directions of those hidden factors. So first we want to either reorient our data, kind of, or project it, so that we are looking at it from the perspective of, let's say, 10, 20, 30, 40, 50, certain number of, but fewer than 20,000, the number of genes, fewer than the total number of genes. We are going to reduce it to, let's say, 50 dimensions of hidden factors. And that's the goal, to reorient our data in terms of these hidden factors, because those hidden factors are perhaps fewer in number. And if we look at our data in terms of them, then we can have less noise, or and we can enrich for signal, and we can have more computational efficiency, which will be helpful in our downstream steps for some of the purposes, especially clustering. So that's what is going on. And how do you do that? How do you identify, in which direction is the, data most stretched out or spread? Well, there are, as I mentioned, linear algebra techniques and algorithms out there. And well, this algorithm PCA was first developed at the turn of the 20th century. But since then, there are many variations of it. And the classical approach for PCA, it is accurate, but it is also not scalable, because it was not meant to with, it does not scale to the level of or number of dimensions and the scale of data, millions of cells, or thousands or tens of thousands of cells that people are working with. It does not perform computationally as well. It can take a while to perform PCA, if you run classical approaches. So there have been many different optimizations which have traded off a bit of accuracy for computational efficiency. And you can read this benchmarking paper in, for some comparisons. In general, I don't see much discussion around PCA. People have people. I think in Surat provides only one approach for PCA. I don't think there are multiple. Let's just check. I don't think there are multiple versions of PCA that you can select, although you can set some parameters. So PCA is generally considered more stable and robust. So there are, each package has some implementation, but if you want to dig very deep, then you can look into it in more detail. So the goal is here to identify some top or a certain number of components or hidden factors, directions of hidden factors, which are dominating the biology in our system. And we will reorient the cells in terms of those axes, so to say, and look at the data in that space, PCA space, for clustering purposes and nonlinear dimensionality reduction in the next steps. So you can select for top 50 dimensions, but there are also some diagnostic plots or some plots which you can look at, as I will describe in a bit, to identify how many dimensions may be worth considering. So an assumption in all these analysis approaches that we are, or steps that we are implementing, is that the data can indeed be represented in fewer dimensions, and that there are some hidden factors which are dominating the biology. And you can hopefully, your analysis will confirm that, or other approaches will help you validate it. But that is an assumption you make going in. And here is an illustration, another illustration, showing how it may look like if you have, let's say, only 3 genes. Again, that is the maximum we can plot at a time. If we are looking in the gene expression space, then it may so happen that the cells are distributed along a plane, and perpendicular to the plane, there is mostly noisy variation. And along the plane, you see separation of, let's say, the cell types or the clusters of samples or something, diseased versus healthy, or maybe different drug treatments. You see the separation of those cells or samples in the direction of a plane. So if you reorient your cells or samples, in terms of the plane, then you may be able to see the data and the relevant aspect of the data with respect to the plane. And here you see that there are 4 clusters of cells. So again, this is called, we will call it PC space or PCA space. PC space, I guess, is more appropriate. So it is common to work with top 10 to 15 PCs, but we should try to err on the higher side. If we are not very sure how many PCs to select, then select a higher number than lower. And there are exploratory visualizations which we can look at, as we will just do. So I will go to this, I guess. Let's look at our code here in the Markdown HTML file, which was generated by the same Markdown document. So you run PCA on the set of genes which you have selected for. So we selected, first of all, Feature selection is also a kind of dimensionality reduction. From 20,000, we reduced the dimensions or dimensionality of our data to 2,000 dimensions, which was based on the highly variable genes, although this is not what people are typically referring to when they are calling dimensionality reduction. So after this selection of features, we run this algorithm PCA, and it will identify those new directions. And here I have plotted how cells are in the top two principal component space. So you can again, do only 2 or 3 at max if you want to plot, but you can plot it in the second PC2 and PC3. So all the PCs are ranked by their levels of significance, I guess, in for the data, for in terms of explaining the variance observed in your data, or the stretch of the how well the data is stretched. So actually, I forgot to point out that before you do all this, there is also a step of scaling the data which you have to perform, which is which will center all the expression values to 0, and also standardize the variance of the expression levels. This is again done so that you can the highly expressed genes and the variance in highly expressed genes, which can be larger, do not end up dominating the analysis. So there is some standardization going on there upstream. So we can look at the data in 2D, but there are some plots. Let's look at this one first, the elbow plot. What this is giving you is the variability in the direction of different principal components. So each axis that we identify, each new direction we identify, is what we are referring to here as principal component. And in the direction of each of the principal components, there will be some variance. And the most significant one will have the highest variance, and that is what looked like the stretch in our figures, the spread of the data. So if the variance in this direction is very high, that is the first PC, and the second PC is the second direction in which, or the second direction is second PC is the one with the second highest variance in the data cloud. If you want to be more imaginative, data is spread here, and the variation in the second highest direction of variation, or the direction with the second highest variance, is what we are calling the second PC. So you can make this PC plot where each PC has a variance or standard deviation associated with it. And what we are looking at in this elbow plot is the way this variance decreases as we go from one to second PC to third PC to fourth PC. And after a certain while, you will find that the change in the variance or standard deviation from one PC to the next PC is not too much. So for example, here close to 15 or 20, we are seeing that the standard deviation does not vary that much. So maybe we can just work with 20 PCs, because after that maybe it's noise, and that's why there is not too much variation, because all the noisy directions will hopefully have almost equal amount of variance. So that is the kind of assumption here. Does that make sense? You can let's look at another example for the data analyzed by the Surratt authors for their illustration purpose. So here you can see that there is a very long plateau, which means maybe selecting 10 or 15, maybe if we want to err on the higher side. Selecting of more PCs than fewer will not be so harmful as selecting fewer than what is appropriate. So if you have to be more conservative, then err on the higher side. It will come with some additional computational cost, but I think 10, 15, and adding 5 more components will perhaps not be that expensive computationally. There is another kind of plot which you can look at, which is called as a jackstraw plot, which we have also generated, but I think This plot takes a little bit longer, I think, to generate because of some permutation-based test which is going on in the background. So I have not, I think I specified very, I did not run it for, as much as one should perhaps to evaluate all the PCs. What one wants to look at in this jackstraw plot is the p-value associated with each of the principal components. So here on the right, there is for each of the PC, there is a p-value listed. And we can see that when we get close to 13, 14, or 15 PC, our p-values are um, kind of high, so they are not significant. And this is what the p-value is representing. The way it is generated is a bit complex to explain, but it is based on some permutation-based test where they are looking at the association of genes with the principal components. And if there are more genes associated with the principal components, than one would expect by chance, then those principal components are significant. And that is the kind of information that is also given in this plot here on the along the x-axis and the y-axis. What we have is the from a permutation-based test, the p-value for the genes. Each dot here is a gene, so the p-values for the genes from a permutation-based test. And on the y-axis, it's expected value. And if certain genes, if certain PCs are not significant, then they will both the expected value based on a null or on a PC being not significant, and our empirical observation for the PCs will be overlapping. That is to say, the values will be the same, identical, and so the curves or the PCs for which the curves are close to this diagonal line are not significant. That is the interpretation. Any questions on that? So, well, what we have looked at now is what is I mentioned earlier as linear dimensionality reduction. It serves the purpose of denoising the data, and also reducing the dimensions further, which will improve our computational efficiency. But it may not meet the requirements for visualization. And I should emphasize, may not. It depends on what the goals for visualization are. But for certain kinds of analysis or purposes, such as, let's say, we want to see trajectories of how one cell, certain populations of cell differentiated and changed into a certain another cell type. So in those cases, maybe our goals of visualization will not be served by PCA kind of analysis. And the reason for that is what I've tried to illustrate with these figures on the right. So let's say we start with a population of cells which are lying, at this kind of a position, where here I have in this figure a rolled paper kind of a thing. So let's say our starting population of cells, they were located in the gene expression space or in our PC space. They were located at the in this red region of the paper. And as time passed and the cells differentiated, they moved along this non-linear trajectory. They moved from along this path from red to blue to green to yellow, and they ended up here at this point to differentiate into a certain another cell type. So, and this may happen in our system because it is possible that cells will not jump directly from this red region over to the yellow region. And I will explain in the next slides, why that may be the case. But it is a common observation that in biology, nonlinear structures of data arise all the time, and in other fields also. So that's why people have developed these techniques of nonlinear dimensionality reduction. But if we apply a linear projection technique to this kind of data, then we may not be able to capture in our projected projections or visualizations, we may not be able to capture the the way the cells, actually changed their expression levels or positions in the space, or the trajectory of the cells in the space. So here, for example, if we project them linearly in any direction, we will end up with a mixing of cells. So let's say we project it using PCA approach, then the PCA approach will see a lot of variation in this direction, a lot of variation in that direction. So it will perhaps project it such that the green will become closer to the red than the blue. Green is in between the blue and the red part, because that's how the PC, it will look at only straight lines in straight line directions, and the cells get mixed up now. Now, if you want to plot on this in this 2D space, the trajectories, then it is hard to visualize, because the cells from here go here and come back here. Well, that looks, maybe it will not make much sense. So if we want to, or in another way of saying this is that, when we are projecting our data in 2D, this is this kind of this is also called as an embedding of data in 2 dimensions. So from higher dimensions, we of course do not have 3D. We have, if we have done PCA projection into 50 dimensions, and we have 50 dimensions. So from those 50 dimensions, we want to visualize our data, and what we want to preserve is the local neighbor, or the neighbor of each cell. So this cell, any cell which is located here, its neighbors are red cells. And then a cell which is located here, it has maybe some cells which are blue as neighbors, some cells which are red as neighbors, but not the ones which are green. So we want to preserve what people describe as the local structure of the data. Again, using this illustration at the bottom. So let's say we have data distributed along, this kind of data has this structure, S-type thing, where, or cloud of data is forming an S-type structure. So then the local or the neighbor of this particular cell is this cell. The yellow cell has this neighbor and some orange cells. So that's the kind of structure we want to preserve in our 2D projection. Maybe that is our goal. So for that, there are techniques which are called as t-SNE and UMAP. Those are the popular ones in single cell community, but there are of course many others. As anything for any other step, there are many options for dimensionality, nonlinear dimensionality reduction as well. But the goal is that we will have our neighbors preserved in this 2D embedding. And well, before we proceed with how this happens or how the algorithm works, a few notes on why this is commonplace in biology. And one reason is that gene regulatory networks contain feedback loops. So for example, any genes, its expression level cannot just keep on increasing. After it has increased to very high levels, it has to come down, maybe because of negative feedback, or there may be other reasons. So you have all studied those kinds of things, perhaps in other systems or in your own research. So you would know that there are gene expression is generally not linear. As cells evolve, things can go up and down. And also with cell cycle, some expression of certain genes can go up and down. So also similarly, there are other kinds of reasons because of which the cellular trajectories may take nonlinear paths, such as protein-protein and protein-DNA interactions, which may manifest cooperativity, ultra-sensitivity, those kinds of response curves, sigmoidal curves, which you are perhaps all familiar with. And cells also, those studying developmental biology might be familiar with this very famous Waddington landscape figure, where the illustration shows that cells, as they differentiate from one type to another, they cannot just let's say this zygote cell ends up at this location where it is here. Illustration shows adult cancer cell, but it could be some cardiac cell, or let's say neural cell, or something of some other cell type in our body. Then it cannot just fly from there to here in a straight line like a bird. The cells, as they as they differentiate, they observe or they encounter, some inaccessible regions of the gene expression space, and they have to take different directions along their paths of differentiation, which is just another way of saying all these things which I just said earlier. So in general, the bottom line is that the paths of cells are maybe nonlinear. And for example, if data is lying along noisy loops in gene expression space, then it might hint at oscillatory gene expression dynamics. And again, I have some reading suggestions here in case you are interested. You can find some good explanations for why this happens in biology. And here is another example in 2D. So if we are looking at just two genes, let's say, then as development happens, the expression level of one gene goes up and then it goes down, while in the meantime, the expression level of another gene picks up. And if you look at if you look at cells in the space of these two genes and their expression levels, then you will see that the cells are moving along this red curve path where the expression level of A goes up while B stays constant or close to zero, and then A decreases while B picks up. So again, these trajectories of cells, such trajectories are commonplace in biology. And for those purposes, to facilitate visualization of data in presence of such nonlinear dynamics, people have developed t-SNE, UMAP, and variety of approaches, but there have also been questions raised. For example, I have an excerpt here at the bottom, and it's not my opinion. Some people have raised question where it looks more like people have said that it looks more like modern art to please the reviewers and collaborators than actually to contribute anything to science. So some people have claimed that, or raised at least those kinds of concerns. But this is perhaps not entirely valid. So we want to understand what is going on in a little bit more detail, and I hope to answer this question about whether it is art or science. And also this question, which oftentimes comes up, if you run these analysis, or if you run the step of UMAP and t-SNE projection again, then your output may differ from today to tomorrow. So why does that happen? So we want to understand that in a little bit more detail in the upcoming slides. Before we move to t-SNE and UMAP, it is helpful to understand another approach for nonlinear projection, which is not used commonly nowadays, but it is a good bridge for our understanding. It's kind of a conceptual intermediary between, I would say, linear techniques and the nonlinear techniques, as in t-SNE and UMAP. So as I mentioned earlier, PCA was invented at the turn of the 20th century, and the people did realize that in middle of, I guess, 20th century or sometime, people did realize that there is also this analogous question of nonlinear dimensionality reduction. But there was no solution until early 2000s. I think in 2001, when two solutions were proposed and published simultaneously back to back in, I think, Science. One was called as ISOMAP, and that is the approach which I have in the illustration here. So the idea is that if cells are the data cloud, if it is structured in a nonlinear way, then when we are measuring distances between any 2 points or cells, we cannot measure them along straight lines, which is the approach of measuring the classical approach of measuring distance in classical geometry, kind of which we have inherited from the ancient Greeks. Euclid is one of those ancient Greeks who has written one such popular book on geometry, which is kind of well, whatever about his book. In any case, this straight line distance kind of bears his name, Euclidean distance. So if our data cloud is structured in a nonlinear fashion, then we cannot measure, or our assessment of distances along straight lines are not good measurements. Ideally, we would want to measure what is called as the geodesic distance, which follows the the structure of our the space in which the data is actually embedded, the kind of nonlinear topological space in which the data is embedded. But we don't really actually see this space. What we see are these points only. If we have PC, if we are, whether we are looking at our data in the PCA space or in the gene expression space, all we have are these points. We don't see this whole sheet that I have, here in this figure. So the breakthrough in ISOMAP, or the advance, was that instead they proposed that instead of looking at the straight line distance, as in the Euclidean distance between any 2 points, what we can do is first of all create a nearest neighbor graph, where we will connect each point or each cell with its 5 or 6 or certain number of nearest neighbors, let's say 10 nearest neighbors. And then we will measure the distance along the graph. So between any 2 points, what is their minimum distance? It is the shortest distance along the graph after we have built this nearest neighbor graph. And this shortest distance is a good approximation of the geodesic distance. And then once we have assessed the, instead of Euclidean distances, once we have assessed the distance in terms of the graph, the nearest neighbor graph, then we can do our then we can apply the same linear projection techniques as we in PCA, etc. We can apply those techniques to in conjunction with the approximate geodesic distance using the graph, and together they will give us a nonlinear projection. So that was the advance in ISOMAP, and we will discuss now the UMAP and t-SNE. But before that, I just want to quickly check if things have made sense thus far, or if you have any questions. Again, just to let you know, all questions are welcome. Okay, well, if sounds like you all followed along, that's what the silence sounds like. Well, okay. So t-SNE, UMAP, they are another, they advance the field further by further refining and extending the approach of nearest neighbor graph kind of thing. Well, the one of the problems or drawbacks of this kind of a neighbor graph approach is we are classifying every pairs of points in a binary fashion as either a neighbor or not a neighbor. We are saying, let's say, we say we are going to build a 6 nearest neighbor graph. So we connect every cell to its 6th nearest neighbor. And let's say the 6th nearest neighbor is 10 units of distance away in the Euclidean space. So then what we are saying is that if there is a 7th cell, which is 10.1 units away, that is not a neighbor. So very abruptly from being a neighbor, we have shifted to not being a neighbor, while the 7th point is very close. So, that is what t-SNE and UMAP, they make that transition from being a neighbor to not a neighbor much more smooth. And that is what is illustrated here in the top right. So on this slide, I just want to convey the principle underlying t-SNE and UMAP. If you have this kind of a neighbor graph approach, then what you are saying is that up to a certain distance, everything is a neighbor. So probability of being a neighbor is constant. And after that, nothing is a neighbor. So the probability is basically 0. What UMAP, t-SNE, SNE, others do, they instead of working with a nearest neighbor graph, they convert the Euclidean distances themselves into another notion of distance, which is based on the probability of being a neighbor. So they use a Gaussian kernel, like t-SNE uses Gaussian kernel, which is basically the 1/2 of the the bell curve for normal distribution, which you have all seen. So it converts using this kernel function every Euclidean distance into a probability of being a neighbor. And you can look up math a little bit more in detail if you are interested using the references in the descriptions. But if 2 points are very close, then they have a very high probability, in this new notion of distance, or not the distance in the new notion of distance in t-SNE and UMAP that they are working with is the probability of being a neighbor. So if 2 points are very close, they are very likely to be a neighbor. And if they are very far away, they are unlikely to be a neighbor. And in between, we have a smooth transition from being a neighbor to not being a neighbor. But since we do not have a nearest neighbor graph anymore, we cannot also calculate a shortest path distance anymore. What we have between any 2 points is their probability of being a neighbor or not being a neighbor. So our linear algebra techniques of also do not work here that work for isomap. So they also had to come up with another way of doing the projection. So now using this probability of being a neighbor, we have a new notion of distance which works, or which is perhaps more accurate than the Euclidean distances. At least it captures the notion, it captures the nearest neighbor graph. But to do the embedding in 2D or 3D, what we have to do now is we have to initialize with something, some way of putting the points. So commonly, people would put those cells randomly in 2D. That is our starting point. And then we will optimize our embedding to preserve this new notion of distance that we have just defined in terms of probability of being a neighbor. And we will discuss the optimization algorithm in the slides to come. And that is what these different approaches are doing: SNE, t-SNE, and UMAP. So SNE was the first approach which was published. It's called as stochastic neighbor embedding, where this approach of probability of being a neighbor was introduced. And t-SNE is an extension of SNE. Basically, the realization in t-SNE was that when we project the cells, if we use the same kind of notion of distance, in higher dimensions and in lower dimensions, and we want to preserve them identically, then a problem of crowding appears, which is, in the sense that in higher dimensions, there is a lot more volume than in 2D or 3D. So any cell can have a lot more neighbors in 2D, higher dimensions than it can in 2D without our plots looking crowded. So that is the kind of problem being solved in t-SNE. And basically what t-SNE says is that, in higher dimensions, we will measure distance using this black solid line as in the bottom right curve, while in lower dimensions, we will make things more spread out. So the probability of being a neighbor decays a little bit, less rapidly. So with that distance, there is more increased room in the 2D, and data can be, the crowding problem is better addressed. So once again, another illustration to show you what these different algorithms are seeing. So PCA, if you have cells located in different directions, because PCA considers all pairwise distances equally, it will see this dense ball. While t-SNE and UMAP, since they are working with this different notion of distance, from this cell to that cell, effectively this cell and those cells are infinitely far away. So you don't see t-SNE and UMAP don't see them, don't see a link between them, kind of. While points which are close together, they have a link, and the links get weaker and weaker as the points get further away. So with that kind of approach, t-SNE and UMAP, they can preserve the local structure of the data and can unroll things like, the surface in 2D, where blue points are together and closer to the yellow points than they are to the red points in our 2D embedding. So how do they do that? Well, in all these optimization algorithms, there has to be a cost function which the algorithm will look at. So we have a distance assessment in higher dimensions in terms of probability of being a neighbor. And here, these are all illustrative values. These are not what the real values are. I just punched in some numbers randomly. Perhaps they could be better numbers. But in any case, we will have, let's say, some observation of distances, which we can, in higher dimensions, we do have the data, we have their location of the cells. So we can calculate the distances. This is known. And we have to find what is the optimal location of those 2 points in lower dimensions. So let's say we start with some value, and then there is a cost function, which I will just illustrate in a bit, which will describe what is the cost of there being this discrepancy. These two distances are not exactly the same. So this discrepancy has a cost. And if there is a high discrepancy, then the cost will be higher. And we will add up the cost for all pairs of cells and try to minimize this cost for our entire population. And this is how the cost function looks for UMAP and t-SNE. On top, we have the cost function for UMAP. So let's say we have a pair of cells which have distance very low, which are very close together in higher dimensions in terms of our probability of being a neighbor. And so this axis, along this axis, their distance is located somewhere here. And we want to find where should we place these two cells in 2D so that cost is minimized. So let's say we start with the two cells being somewhere here. We place them far away. Our initialization was random, so they could be anywhere. Let's say they were very far away. Cells which were close together, they end up being very far away. So the cost of that will be it is along the y-axis, along the z-axis, or labeled here as cross entropy. So let's say the cost is somewhere over at this level. So what the algorithm will try to do is move towards this valley in our plot, or the lowest minimization. It will try to minimize this cost, and there are it will basically look at the gradient of the cost function at the point of current 2D distance and the actual actual high dimensional distance, and it will try to reduce it so that these cells end up close together also in 2D. It will search for this valley, and it will end up it will try to push the cells close together. That is basically what will happen. And similarly, if two cells are very far away, then the cost of being very close is kind of high. So if the cells in our initialization were very close and in higher dimensions they were far away, then the algorithm will again try to move it towards the minimum, which is again along this valley. So that's how, but this algorithm is working on tens of thousands of cells together simultaneously. So there will be some trade-offs. Some cells may not be placed accurately or appropriately in close to their neighbors in the interest of reducing the cost for some other pairs of cells. So this is an optimization algorithm, and it depends on how many iterations of optimization you do, what size of steps the algorithm takes. So when it sees that the cost is very high and the gradient there is a certain gradient towards the valley, then you can also set how large of a step it will take in the direction of the valley in trying to reduce the distance in the 2D embedding and things like that. So the difference between one difference between UMAP and t-SNE is that they have different cost functions. And the cost function of t-SNE is prioritizing keeping cells that are close together in high dimensions, close together in lower dimensions, but for cells which are far apart in higher dimensions, they can be essentially anywhere in 2D because of the flatness of the cost function for high cost for high distances in the higher dimensions. I'll let you all digest all that. If you have any question, please feel free to ask now. Okay. So because of these, this difference where t-SNE cost function is essentially flat for higher for these points which are far away in higher dimensions. It is claimed that UMAP is better at preserving global structure because it is looking at both, it is trying to keep points which are close together close and far away far, while t-SNE is focusing on the points which are close together and trying to keep them close. So it is claimed that UMAP is better at preserving global structure than t-SNE. However, there have also been some recent reports. So, for example, there is this paper by Kobach and Burens. It's called "The Art of t-SNE" for single cell transcriptomics or something like that. Um, they showed that it is not the default UMAP is not necessarily better than default t-SNE. It depends on various parameter settings, and there are many parameters which you can tune, and depending on the tuning of parameters, we will see different results. And we have to be cautious about what parameters we are setting and what initialization we are using. So I mentioned that commonly people, including in Surat, what we use is a random initialization. But instead of a random initialization, we can use more informative initialization, such as, for example, we can start with the top two PCs. We can have the cells located according to their top two principal components, their coordinates in the top two PC space, and that can be fed to the t-SNE or UMAP algorithms, and that can lead to better preservation of the global structure, because the global structure will kind of trickle through iterations of optimization and be also preserved in some way in the final embedding that we will end after our iterations of optimization. So the optimization algorithm is focusing on local structure. But if we start with something which preserves the global structure, then that global structure will trickle down, and it will be traded off. Some of it will go away. The global structure will, of course, be distorted in the interest of preserving the local structure also. But all this depends on the hyperparameter tuning and settings, etc. So by default, Surat is running Barnes-Hurt t-SNE, which is a bit slower. That is another criticism of t-SNE, that it is slower than UMAP. But that drawback has been addressed in more recent developments of implementations of t-SNE, such as FitSNE, which is available in Surat, but you have to choose it. By default, we have been running UMAP in the core and single cell field. Also, a lot of folks have switched to using UMAP. But you have to be cautious about what initializations you are using, etc. And all that takes more time. So when we get a project, we are oftentimes, well, if you have collaborated with us, you would know that we provide a quote, and given how many hours the researcher agrees for us to work, we have to generate all the analysis or do analysis within those time limitations and budget constraints, and tuning all these parameters take also a lot more time. So we do not we have in all the projects that we have done, we have not always tuned these parameters, which is it is kind of it bothers us, I guess, but well, there are budgetary constraints and practical limitations, because of which in many studies you will find that folks are oftentimes running with default parameters and not tuning stuff. But there are recommendations for tuning these parameters, including in this paper by Kobach et al. And in particular, I recommend actually reading the papers for how to set those parameters. But you want to there are one of the parameters in both t-SNE and UMAP is how many neighbors kind of these algorithms feel for each of the cell. So I told you that they are both based on UMAP t-SNE. They are both based on a neighbor graph, kind of nearest neighbor graph kind of approach. they are doing it implicitly without building the nearest neighbor graph. And when we discussed ISOMAP, I told you that in building that nearest neighbor graph, we have to specify how many nearest neighbor each cell would have. There is a similar kind of concept which is implicitly there in t-SNE and UMAP in parameters. So one parameter in t-SNE is called as perplexity, which sets how many neighbors kind of each cell feels. And similarly in UMAP, there is a parameter called as n neighbors, which is how many neighbors a cell feels. So if you set it to the number of cells in your data, let's say you have 10,000 cells, so you set it to 10,000, then basically you will be running PCA type thing, because all cells will be each other's neighbors. And you will be working on global structure instead of local structure. So that's what it means. And then you can look at more, testing of different hyperparameter settings in this paper, including perplexity, the parameter I just mentioned for t-SNE, and also learning rate, which is about the optimization. As I just mentioned, when the algorithm sees a gradient, how long of a step it takes. So that is what kind of the learning rate represents. Okay, actually, before we move to this, I want to show you something here also, I think here. So here is, you can find also interactive visualizations online, but just to give you a better feel for how tuning these parameters can change the embedding you get. So let's say our 3D data is arranged like this organism or whatever beast, and we want to project its cells in 2D. Then if our nearest neighbors, if our nearest neighbors is very low, that is to say, let's say each cell feels only 3 neighbors, then we are focusing on the local structure, and that's what we see. The red region is close to it, the other red points, and the back green is here, and the legs are here, things like that. And as we increase the nearest neighbors, then we focus more on the global structure. So the horns are far away from the feet and things like that. And also we can control this other parameter. These are the two most commonly tuned parameters in UMAP. So this parameter of minimum distance, it tunes how loosely or densely the clusters or points will be packed together. So if you increase it, then the cells have more room to, the minimum distance can be higher, which means basically that the clusters or cells can be further apart. So you can explore it. You have the link in the slides and with more reading suggestions. So a few final notes on this. I also mentioned that the results will be they have a random component. If you use PCA initialization with t-SNE, then the results can be made deterministic, because initialization is no more random. And if you do affix the number of iterations that will happen in t-SNE and all the parameters, then you will get a fixed output all the time. But this may not be true for UMAP, because the optimization approach in UMAP is also has a kind of a random component to it. So even if you use an informative initialization such as PCA or something, then UMAP may give you random outputs. So you want to save your results after you have decided that this is the UMAP that I want to work with. You should save your output. And you should also save the seed used to initialize the random number generator in when you are working with UMAP or t-SNE, and save the output, of course. Additionally, what you can do is another recommendation, or if I have to give, is in addition to the UMAP plots, you can also have another plot, such as MDS plot of the cluster centers. So you have, instead of plotting So alongside the UMAP plot, which has each cell, each point here is a cell, you can have another plot which is called as an MDS plot. It is a technique similar to PCA, and or you can also use PCA, but basically what you can work with is the centers of each of the clusters, the cluster mean or cluster median, and you can just show where these cluster medians or means are in relationship to each other in another plot. And in that plot, you can focus more on the global structure. So MDS and PCA, since they are working on global structure, so this plot will indicate the global relationships between different clusters, and another plot will indicate where the structure within a cluster, the local structure of the clusters. And I think I mentioned this perhaps briefly, but we can, if we in a UMAP or t-SNE plot, we should not try to read too much about the distances between different clusters. So if two clusters are close together, then another pair of clusters, it does not mean that they are more similar than the other clusters. We should be cautious about making those kinds of interpretations. And finally, another good practice might be to quantify the level of distortion. So another study, which is I think still on bioRxiv, it reported that all this opening of the nonlinear structure in 2D and flattening it out, it can be done if a flat structure actually is possible, but it may not be possible. For example, a sphere, whatever you do, if you project it in 2D, you will be distorting the structure. There is no way, with this S curve, we can open it up and lay it out like this, but with a sphere, there is no way you can do it without distorting the structure somehow. So, it may be good to quantify the distortion that is there in the data in terms of how many nearest neighbors are preserved and how many nearest class means are preserved from high dimensions to lower dimensions and things like that. And you can look at those kinds of things. And at the bottom line is, when you see things located here and there, random populations, then you can ask, what is this? Is this a manifestation of the optimization algorithm not giving you a finally good conclusion, or are these rare cell types or distortion or doublets or dead cells or empty drops? What are they? So you should confirm and validate whatever you conclusions basically these plots are meant for hypothesis generation. They are not for making conclusions. Whatever conclusions you want to make, you have to either go back to higher dimensions to confirm that there is some agreement, or do some other low throughput methods maybe to test your hypothesis, things like that. And then there are additional methods for also PCA people have discussed, and there are lots of variants. For PCA also there are how do you measure variance? PCA is trying to find the directions with the highest variance, but what is a measure of variance? How do you measure that? And if there are batch effects also present in the data, how do you reorient your data so that the batch effects are also accounted for? So basically this whole field is also, there are so many different algorithms out there. And that's why the conclusion is that there is science behind dimensionality reduction, and it is useful for scientific purposes. But there is also an art component to it. And the way I think about it is, if we are doing microscopy, then we may have to arrange a bunch of lenses in certain way so that we can capture the kind of images that we are looking at and focus on the things that we want to focus on. And perhaps good photographers amongst you will know all about tuning cameras here and there. But anyway, similarly with the dimensionality reduction, these algorithms, they are the kind of lenses which are allowing us to explore the data which actually exists in higher dimensions and explore it in 2D or 3D at max. That's how far we can go with our vision. And we can apply these algorithms, and just like we would have multiple lenses in microscopy, we might want to have multiple such steps of these algorithms to get us the kind of view we want to get of the data. and that's the art of dimensionality reduction. So, well, that was, now we have one more topic to discuss, which is clustering. I think that won't take much time. So if you have any question regarding dimensionality reduction or things we have discussed thus far, please feel free to ask now. So finally, after we have done, dimensionality reduction, there were two kinds of the dimensionality reduction we discussed. One was linear dimensionality reduction, PCA. And then after we have identified the PC coordinates of the cells, then we feed it to the UMAP or t-SNE algorithm to give us a 2D view of the data. And we also have the PC coordinates of the cells. So clustering happens in the PC space, not in the t-SNE or UMAP space, because the data is very distorted in that space. So if you do clustering in that space, then you may find clusters which are not perhaps which may not be biologically relevant or meaningful. So the clustering is done in the PC space. And the assumption is that cells, they are attracted to certain attractor states. Each cell type has a state to which it is trying kind of it is being attracted to. If you well, you can read more in literature, but there are some stable states. Maybe cells are moving towards those stable cell types or stable expression levels of genes. Maybe cells are moving towards them. They may not have them, but they are moving towards them. So they are limited or confined certain cell types. The cell types will be confined to the basins of attraction of their defining state, kind of. So that is why clusters arise in the data. And our goal in clustering is to identify those basins. Since we don't know all about the mechanistic details of the biology, we cannot theoretically know how those basins are, where these boundaries separating the different basins of attractions of cells are. So we have to rely on unsupervised approaches such as clustering or unsupervised clustering. And one common algorithm, which many of you may be familiar with, is k-means clustering. That is not the preferred approach for clustering in single cell data. And that is because the structure of the data, as we discussed, it can be complex. And in general, k-means, the classical k-means, it assumes that the clusters have a spherical or ellipsoidal kind of space. I think it assumes a spherical structure of the data. So the data is, kind of, if it were actually in balls, then it will be able to identify them. But if you give it complex structure of data, then it will still be identifying, balls together. So it will focus on clusters will look more like balls in the whatever space you give it. So that's what is being shown here. If you do k-means on the raw data, then you identify clusters which have a mix-up of cell types. And also, if you do k-means on top 30 PCs, again, you have some mix-up of cell types. Less because PCs have less noise, but still it's not appropriate. So the approach for clustering in single cell again relies on the neighbor graph kind of approach that we just discussed. One again builds a neighbor graph, a nearest neighbor graph, and that is done in the step of find clusters. First of all, you create the neighbor graph, find neighbors, and then you run the find clusters function to generate the cluster. So find neighbors will take an input, which will be the number of dimensions of the PC coordinates to use. So we have done PCA reduction of the data, and we can specify how many dimensions from the PCA space we want to use to build our neighbor graph. And that should be based on our assessment of the elbow plot or Dijkstra plot, as we discussed. It could be 20, 30, 40, 50. should be a judicious choice and not too low. And after you have done that, then the next step is building, finding clusters in this neighbor graph. And for that, the approach used in single cell RNA-seq commonly is one of the approaches is called as Louvain algorithm. Louvain is trying to find communities or clusters of cells such that the uh, edges between the within the community are more, or the cells are more connected within a community than they are with cells outside the community. So for example, here, let's say, there are lots of edges within cells in this region, and there are only a few links with this next region. So maybe this is one community and we can call it one cluster. And there is this another cluster. So that first you build the graph, and then on the graph, you do what is called as modularity optimization. Again, there is an optimization routine where what we are trying to do is this time maximize a function where the function is basically looking at the number of edges within a community. So this EC is the number of edges within a community, and this KC square by 2m, this is the expected level of edges by chance. KC is the sum of the degree of nodes, and 2m, the m is the number of edges in the total graph. There is a parameter gamma, which we can tune. So if we set this to be high, then we will get more communities because it will impact the modularity gain function, and which we are trying to maximize. So that is the parameter that we are tuning here in this R script with the resolution parameter. So you can set it to a higher value if you want more clusters, or if you feel that there should be more clusters because you know how many cell types are there in your data. So maybe you know that, based on your knowledge of how many cell types were present in your single cell population, you can tune this parameter to get a reasonable number of clusters. That is the kind of approach in the field now. But there are, of course, also, there are always some statistical tests which are based on, comparing differences between clusters and things like that. Like when the differences between clusters plateau out, then that's the level of resolution you set. And there are other ways that people do it, but we don't have time to discuss all that. In Surat, there are several clustering algorithms available. Louvain is by default the choice, and in practice, it works well. People have raised certain concerns about Louvain, but I think they may not be so serious of a concern in single cell biology. That's why Louvain remains the default still. The way this modularity optimization happens is, in every round, the algorithm will take one cell from what is previously called a community and assign it to another community. And it will check what is now the modularity gain. And if there is an improvement in modularity, then it will keep the assignment. Otherwise, it will revert. So that's the way it happens. And folks have pointed out that in doing so, it may happen that, one node which is bridging between two parts of the community. So there is this node, for example, which is a bridge between these two parts. It may get assigned to another community, and then we will have clusters which are internally disconnected. So there is another algorithm, Leiden algorithm, which addresses that and guarantees that the clusters will be internally connected. But I think that may not be such a big concern in single cell data. However, you do have the option to run it in Surat. And this is the final slide. And after that, I will point out some more things which are not necessary for our discussion, but will be good for your knowledge. So after you have done clustering, it may so end up that, using Louvain or Leiden or whatever else, you end up under clustering or over clustering. So for example, you can tune the resolution parameter to control that, but you may identify some clusters which you feel like, after you have done some analysis and maybe even written a paper and your reviewers comments come back, and you find that the reviewer pointed to a cluster and said that it seems like that cluster could be broken up into 2 different clusters, and you want to do further analysis. And now if you tune the resolution parameter, then it will impact all of your analysis. Instead of that, what you can do perhaps is look at one specific cluster, take its cells, and do another iteration of clustering on just this subset of cells. So that is an allowed approach, and you have some references here in case it helps you. So I think those are all the things we wanted to discuss. Fine. For marker genes, you can again, after you have done the identification of clusters, you can run the find markers functions from Surat. You can read more about it in your own time. Ruben will be discussing differential analysis and comparative analysis of cells on tomorrow. So I hope you will all join us again tomorrow. And here you can specify which 2 sets of cells you want to compare. And here I have given some examples, but there will be more discussion on this tomorrow. Also examples of other plots that you can generate. Well, I will answer some questions now, but before that, I will point out one thing regarding our clustering and UMAP plots. One thing you can see, I have run both UMAP, t-SNE, and PCA. You can compare these for how the clusters are located, or how the cell clusters are located in these 3 spaces. In PC space, we can see that many clusters have gotten together and are overlapping, which is because this is here global structure dominates. In both the UMAP space and the t-SNE space, they both have the same cluster labels. All cells have the same cluster labels, and we can see that the clusters are in separate locations. UMAP plots are more tightly knit or tightly packed because of the parameter settings. You can tune the parameters of t-SNE also to get tightly knit clusters here. But few things you can note. So for example, 1, 2, and 16 are here like this. 1, 2, and 16. And 1, 2, and 16 are also in kind of similar structure in the UMAP space. So there is some correspondence between the 2 plots, but well, there are also differences, which are because of reasons as I explained. So I hope all these things and our discussions made sense, and I hope I was able to give you something useful. But now I can take more questions, and that's all I had to share with you in this session. Thank you very much all for your participation, and I hope that you will all take the time to provide feedback. Without your feedback, we cannot improve and provide these workshops or in a meaningful or useful way. So please do provide feedback. Okay, now I will answer some any questions that you may have. Please feel free to unmute yourself and ask them. Here we have one in the chat box from Wanping Wang. If I have gene of interest, what shows a trend? If I have a gene of interest that shows a trend in developmental trajectory, how do I find other genes showing a similar expression profile? Well, we have not discussed a trajectory analysis, but if you have a developmental trajectory which you have done using any tool such as Monocle or Slingshot or or there are so many tools out there. So those tools will generally also give you a pseudo time assessment for the cells, and then you can look at the genes which are correlated with the pseudo time trajectory for the gene of interest. So genes that are along the same trajectory path will have correlation with the pseudo time that these tools will give you, and based on those correlations you should be able to identify. That will be my first guess, but there may be other approaches. Ruben, Min, Alex, anyone else have any answers, please feel free to also chime in, or anyone else among our attendees if you have more experience with trajectory analysis also.
Yeah, I would, this is Ruben here. I would just maybe, because seems like there is a given gene of interest and you want to find out other genes whose expression is associated with that gene of interest. So another way of saying it is maybe you want to find another gene whose expression is correlated with this gene of interest. So then something as simple as a correlation, look for correlations between expression of this gene with other genes, maybe in particular clusters of interest. You just pick expression of this gene in, let's say you're interested in cluster 1 and cluster 2, which represents maybe 2 endpoints of a trajectory. Just look at, estimate the correlation coefficient. It would be a simple analysis.
Thanks, Ruben. We have another question for, and I hope that was satisfactory answer for you, Wenping. So we have another question from Aminoli. Oh, I missed the part when you said about clustering. What are the methods of finding the clustering, or whether it is over or under clustered? So I did not mention anything about finding whether a cluster is over or under clustered. My understanding is generally that happens based on our biological understanding. So for example, you have a cluster and you try to find marker genes for those clusters, and you get a result from your analysis of find markers or comparative analysis of clusters, but you cannot identify, you don't know of any cell type which has those marker genes. So then a reasonable conclusion there would be that this cluster is not one cell type, but perhaps a mix-up of cell types. So maybe there is some point in doing reclustering. So that may be an indication of over or under clustering. Or there are, of course, there are other approaches where what they do, the statistical or data-driven approaches, where you test basically a bunch of resolution parameters, and you look at the within cluster similarity and the differences between clusters, and you will basically, just like in our elbow plot, you will find that the whatever functions you may implement to assess within group similarity and within cluster similarity of cells and the between cluster similarity of cells, you will find that there is a plateauing out where after increasing or decreasing the resolution parameter, the within cluster similarity does not improve, or between cluster similarity does not it should be high, between cluster similarity, sorry, should be low. So it also does not increase, and the within cluster similarity also kind of looks like it is optimum. So that based on those kinds of approaches, you can also determine a clustering parameter, a resolution parameter for clustering. Any other insights on that topic also? Anyone? I hope that
Hi,
was.
just wondering with regards to following up from your explanation just now, what's the name of the statistical method to find if the clusters are under or over, like you were saying just now? Is there a specific method, or
Yeah, there is one. I forgot its name. Do you know, Ruben? I have not read it in many, certainly many months. So do you know, Ruben?
Yeah, yeah, the actual name escapes me too, but I think the way you describe it seems that's what you're looking at. I think one approach, at least when one thinks of k-means clustering, plot, a term which you've already encountered in Krishna's presentation today, is called elbow plot.
Yeah.
Similarly, there is an elbow plot where the x-axis, or sorry, the y-axis would be that between versus within cluster differences.
Perfect. Thank you.
Yeah.
Any other questions?
I have a quick question.
Go ahead.
If we wanted to iteratively search for hyperparameters such as the resolution or the number of k-nearest neighbors, what do you think is a good metric to measure to implement that, like a cluster variance or?
Yeah, similar kind of approaches that we just discussed, for clustering, I think, yeah, variance within a cluster, between cluster, can look at those kinds of things and summarize. I have not done that much, or it's been many months since I've read something like that, so I cannot give you precise recommendation. But if you look for in the literature for optimization of cluster resolution parameter, clustering resolution parameter, I'm sure you will find several options.
So if you have multiple replicates in your data, that is, your cells come from different animals or different individuals, then in my mind, the ideal way to be able to optimize for these hyperparameters would be to do a cross-validation kind of procedure where you fit your data for whatever resolution parameter you pick on a subset of cells, on a set of cells from a subset of samples, and test how well the resulting cluster is on the set of cells that you haven't used to build that model for clustering. So yeah, that would be an ideal approach in my mind if you really had enough replicates, biological replicates, not number of cells, but different biological replicates.
So I just shared a link for a paper describing what is called as the gap statistic, and that is the one I was referring to, for optimization of cluster, the parameters.
Thank you very much.
It has perhaps been implemented for some single cell RNA-seq data also, but I've not done much of, honestly, clustering resolution optimization. So I can only point to my literature study. Any other questions? Okay, folks, thank you very much for taking part in our workshop, and we have another session tomorrow and the day after. So we hope to see you there.